Okay, first I'd like to thank Karen and Eric for inviting me and for the opportunity to speak. I'm going to be switching gears in a number of ways right now, so hold on, everybody. One of the topics I'm going to discuss is sleep, and I hope we're sort of past the postprandial somnolescent period. That was during the last two talks, and everybody's wide awake now, right? So in any case, we're going to be talking about neurons, we're going to be talking about circuits, and we're going to be talking about epilepsy. So three changes in direction from what we've discussed so far. You all appreciate that seizures are a significant outcome in GBM, and people like Harry Sonheimer have studied this extensively in terms of the types of changes that happen in the peritumor area, including changes in glutamate uptake, changes in chloride ion channels, et cetera. There's potential interventional therapies to be developed there. But I'm going to be talking about the circuit. So what happens in epileptic brain? What are the changes in the brain that occur that lead to seizures? And I hope it's informative to the GBM field. So with that, here's an overview of my talk. I'm going to be talking about the thalamocortical system. We've heard a little bit about the thalamus and how it's a dangerous area to go after surgically. Personally, I'd be really offended if you took my thalamus out. I think I'd lose consciousness, among other things. But the thalamocortical system is a key neural circuitry involved in a number of seizures, including absence seizures and other forms of epilepsy. I'm going to talk about the critical elements in the circuit, in the thalamocortical circuit, that are involved in seizure generation, and how we can get clues about which cells and which synapses are important for the seizures. And then I'm going to discuss a study we recently published showing abnormal thalamic function after a focal cortical stroke model. And so this may intersect a little bit with the types of injuries and reactions that are happening in GBM. And finally, I'm going to talk about our most recent studies to target the thalamus with optogenetics for real-time seizure control. And I think the message there is that the barbarians are not going to go out of business anytime soon. There's going to be many new interventional approaches if we can work out the kinks, as it were, with viral gene therapy and intracranial device implantation. But I think I'm optimistic that those things can be solved. Okay. So the thalamocortical system and epilepsy. This is a slide that we saw in the middle of the last presentation. Petit mal epilepsy is a form of epilepsy that happens mainly in children. It is characterized by this highly synchronized EEG activity, so multiple electrodes on the scalp EEG, so this sudden and massive degree of synchronization of neural firing. So the brain switches its state quite suddenly from normal behavior where there's this low amplitude, high frequency activity to this very high amplitude and low frequency activity. And it's really striking. If you see a patient that has an absence attack, they will be talking and then suddenly they'll freeze. They might blink their eyes a little bit. The seizure will go on for a while. And then the seizure suddenly stops and the patient will resume what they're doing. So it's as though the pause button has been pushed on their consciousness. So this type of epilepsy strongly involves the thalamocortical system. And so this was first appreciated from depth electrodes recordings by the neurosurgeon Williams in the 50s, where they dropped a depth electrode deep into the thalamus and recorded simultaneous electrical activity along with the cortical EEG. And here you can see this massive wave formation in the thalamus at the same time as the cortical EEG. And more recently, this has been established in a number of neuroimaging studies. This is fMRI staining, showing the thalamus has a high degree of increase in blood flow, therefore increased metabolic state during the seizures. Strikingly, the cortex is, if anything, shows a decrease in metabolism. So during this massive synchrony that's recorded in the cortical leads, there's an overall decrease in cortical activity. So so what is the circuitry that underlies this type of activity? And it appears to derive from a physiological circuit that drives sleep rhythm called sleep spindles. And you all appreciate that sleep spindles are a hallmark of early stage sleep, stage 2 sleep. These are now EEG recordings, and you can see these little waves on the EEG. These are recorded on multiple electrodes, just like the spike and wave seizures that we saw in the absence patient. They have a particular spectral component, so they're high frequency, approximately 12 to 14 hertz, and they're distributed across multiple cortical electrodes. So the sleep spindle is a distributed rhythm that's occurring within the thalamocortical circuitry. And it has this kind of start and stop over the course of about a second. Now, in contrast, this is now recorded in the same patient. This is now this spike wave discharge that occurs during the absence seizure. And what's been redrawn here is the vertical calibration on the EEG. You can see that the local EEG spikes are much greater in amplitude, 300 or 400 microvolts compared to the 50 to 100 during the spindle. So that indicates a massive increase in synchrony. Local neurons are all firing together during the seizure. And furthermore, there's massive synchrony across electrodes. And you can see that the spikes on the EEG are occurring simultaneously on all the electrodes, which is different than the distributed nature, the sparse oscillation that we see during the spindles. So our current working theory is that sleep spindles can interconvert to spike wave discharges through changes in this basic underlying thalamocortical circuitry. Now, one piece of evidence from this is from animal work. This is the model of feline induced generalized epilepsy. A massive dose of penicillin is injected intramuscularly into a cat. And this cat was sleeping. These are barbiturate-induced sleep spindles. You can see a spindle and a spindle. And then over 10, 20, 30, 40 minutes, these spindles are converted to these higher amplitude, just like we showed in the EEG before, and lower frequency activities. So this just suggests that these two can interconvert. And you can even see, here's an event that started as a spindle and then converted into this higher amplitude, lower frequency activity. So that's the thalamocortical circuit. It has a normal function in driving spindles, but we think it also can be corrupted in the induction of seizures. So how might that interconversion work? How can we go from this normal rhythm to this pathological rhythm? So we're going to dive deeply now into some of the physiology, the electrophysiology of these cells and the neurons that are involved. This is a cartoon that derives from an experiment that Mircea Steriad did with his colleagues at University of Laval, involving recordings from neurons in the cerebral cortex and then deep in the brain in the thalamus. And there's basically three types of cells to consider in this circuitry, this rhythm-generating circuitry. There's cortical cells that are long-range projection cells. They use glutamate as the neurotransmitter, and they transmit that long-range excitation to the thalamus. Similarly, there's thalamocortical relay cells, which are responsible for taking all the sensory information you're receiving. Like right now, my voice and the picture of these slides are coming in through appropriate thalamocortical relay neurons, and then those are also excitatory and projecting back to cortex. And then here in the middle is this, in this mustache-shaped structure, is the thalamic reticular nucleus. And this is the key nucleus that I'm going to focus on today. The reticular nucleus is a intrathalamic nucleus. It's composed of these oblong cells that are entirely inhibitory. So in contrast to these cells in the cerebral cortex and the thalamic relay nuclei, these are inhibitory. So this network has long-range excitation and then local loop excitation and inhibition, and these all come into play in the generation of the seizures. What Steriad's group showed during, again, intracellular recordings from a cat during these spindles was the activity in the cortical thalamic cells, so-called CT cells, reticular thalamic cells, RT cells, and thalamocortical cells. They're all involved. These are intracellular recordings showing spiking occurring in each of these cells during the generation of the spindle. And the rhythm generation circuitry actually occurs within the isolated thalamus. And I'm going to go through this in just a second. It's key to understand this because it's that rhythmic circuitry that's involved in the seizure generation. So if we focus on the intrathalamic part of the network, we have excitatory thalamocortical cells. Did anybody put new batteries in this? Okay. Excitatory thalamocortical cells that are interconnected, reciprocally connected with these inhibitory cells in RT. These two cells, through this local loop recurrent connection in the thalamus, can result in a network oscillation because activity in the reticular cells causes a release of the inhibitory neurotransmitter GABA here. This leads to a membrane hyperpolarization, an IPSP, and these thalamocortical cells have this special feature that when they're inhibited, they fire a post-inhibitory rebound. So unlike what we normally think about in terms of inhibition in the brain as preventing firing, this inhibition actually drives firing. And so in this network, we can have inhibition that results in late delayed firing, which then can lead to reentrance in this network because the firing of these TC cells causes glutamate release. And so we can go back and forth between this intrathalamic network, GABA, glutamate, and we have a network oscillation. Okay, and we know a lot about this. We know a lot about the ion channels that are involved in each step of this. And for example, we know that these channels that underlie this post-inhibitory rebound are T-type calcium channels, and they're susceptible to anti-epileptic drugs that can block the seizures. So we'd like to get to the point of understanding how we can get to a highly synchronized network that occurs during seizures compared to the sparse oscillations that happen during spindles. So I'm gonna describe studies in a mouse model of epilepsy. Just again, to briefly make the point about the involvement of the thalamus in these seizures, this is now a rat model with depth electrode recordings, and you can see here down in the ventral basal complex of the thalamus, there's a high involvement of the spike wave activity during the seizures and not in hippocampus or amygdala. Okay, so this mouse was a particular knockout of an AMPA receptor, so the excitatory receptors that were introduced as part of this talk. This is work from Peter Seeberg's lab that came out just a year or two after the big UCSF Genentech fiasco. Seeberg's group identified and cloned these very important AMPA receptors. These are the receptors, glutamate receptors that mediate fast synaptic transmission in the brain. And what you can note is this is a horizontal section from rat brain and a coronal section. And here's this reticular nucleus. You can see that it only contains the GLUA4 subunit. You can also see this here in the horizontal section. So this part of the brain, these inhibitory cells that are so involved in this rhythm generation, express explicitly this AMPA receptor. And we were able to obtain a mouse that was missing that particular AMPA receptor. And surprisingly, if you've been following along, these animals have seizures. They have absence seizures. They're mouse absence seizures, so they're a little faster in their rhythm generation than in humans. But you can see here's the left frontal and the right frontal EEG and the EMG, and you can see the animal stops behaving. The EMG goes to nothing during this seizure, and it's oscillating at about 8 hertz in this highly synchronized way. So how can it be that these animals have seizures? I've said that excitation of the reticular nucleus is part of the recurrent thalamic loop. So we address this by looking at the electrophysiology of the thalamus. And for these studies, we made isolated brain slices that contained just that loop that I've been describing, the interconnections between the reticular cells and the thalamocortical cells in a brain slice. And these are now multi-unit extracellular recordings to just establish how much excitability there is in the network. And what we found, surprisingly, was the GRIA4 minus mice, the knockout mice, had a much greater intrathalamic excitability. So this network, which we thought depended on inhibition and excitation, and this between these two cells was actually enhanced when we lost excitation of the reticular cells. How can we explain this? So there's two possible places that this defect could occur. It could occur from the output from the cortex, so the CT cells. Now I'm stuck with no lasers. I can use fingers. Did you have another laser? Do you have maybe more batteries for that one? I'm sure there's a Walgreens across the street. That there's two sources of glutamatergic excitation of these cells, the cortical inputs and the thalamic inputs. And perhaps the difference in this epileptogenicity is due to specific localization within these. To study this, we used optogenetics. This technique hasn't been described yet today, so I'm going to briefly go through it. I'm sure many of you have heard it. The idea is to render neurons sensitive to light, and you can do this in two ways. You can make them excited by light or inhibited by light, and that allows us to do circuit dissection and, as I promised, intervention in seizures. So there's two main types of opsins that are used, excitatory and inhibitory opsins. They're ion channels or pumps that sit in the membrane. When activated by light, they will allow ions, charged ions, to flow in and out of the cell to cause excitation, or in the case of NPHR, it's an inhibitory ion opsin that fluxes chloride into the cell. How am I doing on time? 12? Okay, good. Okay. Now, the nice thing about these opsins is they can be activated by light. And here we're looking at the wavelength of light versus the activation. And you can see that they are largely non-overlapping in their spectral characteristics. So you can put both of these proteins into cells if you can get gene therapy working, viral gene therapy. You can take these opsin molecules that are from bacteria, express them in neural membranes, and then make those neurons sensitive to light. If you hit them with blue light, you'll get activation, positive ions flowing in, causing the cells to fire. If you hit them with yellow light, you can activate this pump and have the opposite effect. You pump chloride ions in and cause them to stop firing. So here's a proof of concept experiment. Blue light pulses are causing the cell to fire. Now, when they concurrently apply yellow light, they're reducing the ability of that excitation to cause the cell to fire. So nice bidirectional control, and it's extremely fast. These light pulses and the responses come on in just a few milliseconds, so we can turn on and off neural activity very quickly and reversibly. Okay, so this is the question, what's happening in this circuit? We have excitation coming from cortex and thalamus. Where is the defect? Traditionally, us electrophysiologists have been stuck with pretty crude techniques, and we would do, I guess, what neurosurgeons would do and drop an electrode in the brain and stimulate and hope that you could activate a specific pathway. You can see in this case, we can't specifically activate them because the two fibers are intertwined. So electrical stimulation would activate both of these pathways. But with the viral approach, we can put channel rhodopsin in either the cortical thalamic cells or the thalamocortical cells, and we do this just with a specific injection into the virus into the region of interest. So we can put the opsin in the cortical thalamic cells or thalamocortical cells. And then we take our handy-dandy blue flashlights and activate one pathway or the other to dissect out where the changes are happening in the circuitry. It's kind of a complicated experiment. You have to find a virus that will work well, so we use CAM kinase 2 alpha promoter to drive expression in these excitatory cells. We include YFP, a fluorescent protein that allows us to identify which cells are infected, and then we wait for a long time because it turns out that when you put the virus in the cell body and have the neurons start to express that, it can take many months for the opsins to get down into the axons. So you have to wait for that to happen. And then we made recordings from the reticular cells and looked at the synaptic responses. So here is an experiment where we've injected the virus into the thalamus. You can see the green staining here. So this is, all those thalamocortical cells have now been infected with this virus and protein. So they're marked green because of this presence of YFP, yellow fluorescent protein, but they also express channel rhodopsin. And when we apply blue light to these fibers while recording from one of these cells, we get these beautiful isolated synaptic responses. So now these are very specific. These are the synaptic responses that we get purely from activation of the thalamocortical cells. And I think everybody can already jump to the punchline, and you see that this part of the pathway was completely unaffected. So remember, I said these cells are missing GRIA4, which is one of the major glutamate receptors there. But apparently that particular receptor has no function in the thalamoreticular pathway. I'll just zoom through the rest of this because it just describes the result. And then in this experiment, then we injected the virus into the cortex, into deep layers of cortex, into barrel cortex. And now these are those fibers from the cortex down into the reticular nucleus. You can see them nicely stained with YFP. And unlike the cortical thalamic response, we saw that the response from the cortex was decreased, and decreased by about half. Again, I'm not going to go through all the details, but I will describe how we can integrate these two findings into an understanding of how seizures can occur at this network. So this is an integration study where we've left the circuitry relatively intact, and we're recording now from the thalamocortical cells. And what we've shown is that this input from the cortex to the reticular cells is reduced. And in experiments I didn't show, we know that this direct excitation from cortex to the thalamocortical cells is intact. And in the knockout, we know that this results in a loss of feed-forward inhibition, because these cortical inputs can no longer drive the reticular cells to fire. So normally, in a wild-type animal, when we apply this blue light stimulation to activate the cortical fibers, we give a train of stimuli to mimic what would be happening during a seizure. And you see that the effect that you see on the thalamocortical cells is a very powerful feed-forward inhibition. When we drive these fibers over and over again, you get a little bit of firing of the cell, but that's followed by this long period of inhibition. So the inhibition is occurring because we're recruiting this feed-forward inhibition from the reticular cells. They're strongly activated by cortical input, and now that synapse becomes much more powerful than the direct response, and you just get a single action potential. However, in the knockout animals now, this same input now provides a very strong direct excitation of the thalamocortical cells. So there's been a loss of this feed-forward inhibition. This pathway now becomes extremely potent, and we're driving very strong firing of the thalamocortical cells because we've lost the feed-forward inhibitory response. So this turns out to be key, because normally in wild-type animals, you never see a strong activation of the cortical pathway of the thalamocortical cells, and that has disastrous consequences. We think it's that strong direct activation of the thalamocortical cells that leads to the seizures. Just to prove that this is a loss of feed-forward inhibition, if we do the same experiment here chemically, by blocking the inhibitory receptors here, we just see the same increase in firing. So if we block the GABA A and GABA B receptors here, now this cortical output produces the same type of strong thalamocortical cell activation. So here's a model to describe this. We think normally cortical output strongly activates the reticular cells. That leads to a strong inhibition of the thalamocortical cells, and so this synapse outweighs the direct excitation from cortex. And then because of that, you get little TC cell firing and little recurrent activation in the network. But in the knockout, now this synapse has been specifically weakened. Now cortical output can no longer drive the reticular cells to fire. Instead, we get direct excitation of the thalamocortical cells. Again, this never happens in real life. Okay, thanks, 4 minutes. This never happens in real life. You never get this concurrent activation of the TC cells. And remember now, the TC cell input to the reticular cells is perfectly intact. That one was not affected by the mutation. So now we have a bunch of these cells firing, now causing sudden and synchronous activation of the reticular cells. They all get activated, and now we entrain this intrathalamic network, which then drives the seizure. Okay, so in the last 3 minutes, I will talk about the cortical stroke model. Okay. So this is a focal cortical lesion model, which we use with this photoactivatable rose-bangle dye. Intravenous injection, light exposure right through the skull will cause a focal cortical stroke. And what we've shown is that this can produce seizures. And interestingly, it causes this very focal gliosis, astrocytosis within a small portion of the thalamus in the ventral basal area. We did many intracellular circuit analysis experiments to understand what happens, what are the reactive changes that happen in this part of thalamus, and basically showed that the thalamus becomes hyperexcitable. I'm not going to have any time at all to describe those experiments, so I'll jump to the punchline, which is to just point out that the cells in the thalamus, in this injured thalamus, become hyperexcitable. And that suggests the possibility, so here's the result. So in red here is shown GFAP staining, so this is the reactive astrocytosis we see. The green shows cells that we've recorded from, and when we record from those in this region of astrocytosis, we see a large increase in excitability. And the question is, if we target those cells now with optogenetics, can we block the seizures that ensue in these animals? So here's the circuitry describing the effect. The stroke damages cortex. It damages those thalamocortical cells that project to that region of cortex, as well as a loss of projections from that cortical region down to thalamus. But there's survival in this peristroke region. And it's the interconnections in this peristroke region to the thalamus that we think are driving the resultant seizures. If that's true, if we can inactivate thalamus, we should be able to reduce the intensity of the seizures. Okay, so here's the experiment. Here's a rat. He has a implant installed in his brain, and this is something for the neurosurgeons to think about. I don't know if we're ready for this yet, but this is a fiber optic that's dropping light delivery pipette right down into the thalamus. It's connected to a removable fiber optic, and here's a 8-channel intracranial recording device. So in this case, we're using halo-rhodopsin. We're putting it in those hyperexcitable cells in the thalamus. We show that yellow light in vitro is able to suppress firing. And here's this what we call an optrode. It's the optical fiber, and it has 4 wires sticking out past the end, so we can shine light into the brain, into the VB, and we can record the electrical activity that results. Here is the staining of YFP. So these are those thalamocortical cells that were labeled with YFP, and you can see their fibers project into this peristroke area. So these are the surviving neurons that are in the thalamus that project to this part of cortex where recovery is occurring, where the seizures are also being generated. And we can show that we can suppress the firing of these cells by delivery of yellow light. And most importantly, we show that these animals, when they have these post-stroke seizures, we can interrupt them instantly in real time by light activation down deep into the thalamus. So I will stop there. There's a little bit more, but the point I want to make here is that this has an interesting interventional possibility. These seizures are occurring in cortex, distributed across cortex. As you can see, we're seeing them both on ipsi and contralateral areas. But light delivery into a region of the thalamus, a very small focal part of the brain in which we've induced the expression of this light activatable substance, creates a very powerful means to modulate the activity. So that, to me, creates the interesting idea that if we can find nodes like this, like in the thalamus, that are critically involved in seizures and other neurologic diseases, that we might be able to target them with these small optical fibers, along with gene therapy, because we have to make the cells light sensitive. And at least in examples like this, where those nodes play a critical role, we can very powerfully modulate the seizures and do so in real time, which is the last slide here. So this is a spectrogram showing the EEG with a seizure detector, and there's the little bit of seizure that you see before the light is applied and the seizure is shut down compared to the activity that would occur otherwise. So with that, let me give acknowledgments. Most of this work was done by a talented postdoc in the lab, Jean Paz, collaboration with Carl Dijsselroth, who is one of the key inventors in most of the optogenetic technology, some people, Tom Davidson in his lab, and a few other people in my lab. Thank you for your attention.